So now I'll request Dr. Vikas Kushwa. Yeah, he's available.
Yes, sir, I am available.
Yeah, so he'll be presenting on multivalent peptide vaccines to combat vector-borne infectious diseases. So over to you, doctor.
The slide is visible.
Yes, please go ahead.
Thank you, Dr. Dhananjay and the Longevity Asia for providing me the opportunity to present my work. And my topic is a multivalent peptide vaccine to combat the vector-borne infectious diseases. All of you know that the vector-borne diseases are the disease which is basically caused by the parasites, viruses, and bacteria and transmitted by the vectors. According to WHO in 2024, more than 7 lakh deaths annually occurs by these vector-borne diseases. And these diseases mainly the malaria, dengue, leishmaniasis, filariasis, and much more. The disease is burdened in the tropical and subtropical areas of the world and mainly affected the economically poor peoples and the poorest population of the world. This is a table which is represented the list of the vectors. There's the mosquitoes, snails, flies, ticks, and they cause the different types of the diseases and the what types of the pathogens they spread. If you see the map of the world about the global distribution of these vector-borne diseases, you can see here that most of the world is affected by these vector-borne diseases. Treatment for the vector-borne disease, there are lots of medicines and drugs available in the market. Albendazole, azithromycin, amphotericin, and the ivermectin, DC, chloroquine is available. But parasites and the bacteria have developed some resistance against these drugs. So the researcher and the scientists of the world have moved toward the development of the vaccines. Now there are the few vaccines: malaria vaccines, yellow fever vaccines, Japanese encephalitis vaccines, Dengue vaccines. All these are available in the market for treatment of these diseases. Then why we are going for the multivalent multi-epitope peptide vaccines? All of you know that during the COVID we have take the vaccinations. So the work of the vaccine is that they had to induce our immune system so that our body fight with the pathogens. But during the construction of vaccine, previously we used the attenuated form of the parasite, and sometimes these vaccines had the adverse effect on the body. So the development of the new bioinformatics tool, which is called as the immunoinformatics, we have to select the more immunogenic part of this protein, and we construct the new vaccine construct using these epitopes. And these multi-epitope peptide vaccines are safe and effective, highly specific to the pathogens, induce the higher immune responses, it provides the long-term immunity, avoid unnecessary components, and it also economically usable to all of us. So this is the methodology where I am come step by step. First is our retrieval of the protein sequence. If we are going for the development of the any vaccine, we have to choose the such protein which is immunogenic. Means it has to induce our immune system so that it can fight with our pathogen. So there is the two website basically, NCBI and UniProt. We can go there and download the protein sequence of our gene, and then we have to check the antigenicity of this protein by the software Vaccizin. The threshold value should be more than the 0.5. The value higher than this, we can consider that the protein is the immunogenic. Next is the B cell epitope predictions, in which we have to go for the prediction of the B cell epitopes. The B cells are the cells which are basically used for the production of the antibody. Where is the some software IDB, Cypred, laptops, which is used for the prediction of the B cell epitopes. And here the lower side on the right side, you can see that there are block. There are some protein are selected, which is the probable non-allergen. During the selection of the any epitope, we have to know that your protein or your epitope should be non-allergen. It should not cause any allergy to the host. Next is the MSC1 binding cytotoxic T lymphocyte episode prediction. There is the software which is IgB and NetCTL, which is used for the prediction of the epitopes. MSC2 binding STL epitope, there is the same IDB website and the NetCTL, and the CTL pred software is available, which is used for the prediction of this STL epitope. The value should be more than the 0.5. Then here is the construction of the multi-epitope vaccine. After the prediction of the epitopes, we have to go for the construction, in which we can see here, there CTL epitopes, STL epitopes, and B cell epitopes all are joined by each other through the use of the linkers. At the end of the peptide, we have added some adjuvants which help in the inducing the immunogenicity of our vaccine construct. After the construction of our vaccine construct, we go for the analyzing the immunogenicity, allergenicity, and the physiochemical property of our construct. You use the software Vaccinen for the antigenicity, Allentrop for the allergenicity, and the XPACY for the physiochemical parameters, where we can assess the molecular weight, PI value, amino acid composition, atomic composition, extinction coefficient, estimation half-life, instability index, aliphatic index, and grand average of the heteropathy. After that, we go for the structure modeling. Structural modeling of the vaccine construct. For that, we have to go for the design the secondary structure of the construct. We use the cypret, where you can see there is some yellow color which tell that in the our epitope, in the our vaccine, there is some beta strand, alpha helixes, there is some coils, there are some distorted structures. It tells us about the secondary structure of our construct. Further, we go for the prediction of 3D structure, which is used by the Raptor-X. Further, after the construction of 3D structure, we go for the structure validation of the 3D structure. We use the ProAssay web for the validation of a structure, and use the ProCheck and the ProCheck NMR for the structure validation, where Ramachandran plot is shown by this software, and the value more than 90% is said that your structure is validated and well good. After that, construction of vaccine construct, we know that when we are giving any vaccine to any host, it get bind to the receptor of any cells. Here we perform the molecular docking, binding of our construct with the receptor. Here is the image. You can say that there is the blue color is our receptor, and green color is our receptor, and blue is our vaccine construct. And for that, we use the software PatchDock. After molecular docking, we have to perform the ND simulation, which is to examine the molecular interaction between the ligands and the receptor. Here is the RMSD value, which tell us about the distance between the alpha carbon of the stack protein, and the RMS value, which is for the complex reducible flexibility. How much our vaccine is flexible to get bind to our receptor. Further, in the general, in the daily life, if you are giving any vaccines, the body show the immune response. So after the construction of vaccine construct, we perform the immune stimulation on the software, and we use the software C-IMSIN, where we can see that there is we predict the growth of the B cell population, T regulatory cell population, T helper cell population, T cytotoxic cell population, dendritic cell population, natural killer cell population, antibody production, and the cytokine production. All these are methodology performed on the different software using the immunoinformatics tools. There are some multi-epitope vaccine against the Western disease. You can see that there is a 4 vector multi-epitope vaccine against the malaria, 3 against the filaria, 2 against the schistosomiasis, 1 against the trypanosomiasis. Then we come for the conclusion. This is the reverse vaccinology approach to construct and validate the multi-epitope based vaccine model. Next, this is after the construction of the this vaccine construct and evaluating its immunogenicity on the immuno studies. For the in vitro study, we perform the test on the different cell lines, and in vivo study, we perform the test of these vaccines on the animal model. One of the best thing of this multi vaccine is that it provides the long-lasting immunity with the high complete disease eradication. Thank you very much.
Yeah, thank you, Dr. Vikas, for a very good presentations on the vaccines and its related information.